The main advantage of this culturing technique is that you can obtain virgin copepods while controlling their pairing experience and tracking their development. The following behavior analysis method can help controlled and reproducible experiments on the mate guarding behavior of Tigriopus copepods, which are common zooplankton in rocky tide pools.
Though these methods can provide insight into the reproductive behavior of Tigriopus, they can also be used to look at how genetic and environmental factors impact the physiology and behavior of these copepods.
To begin, use a Pasteur pipette to collect gravid females carrying clear orange eggs from the stock culture. Rinse the females with clean culture medium via gentle pipetting. Next, place each female into an individual well of a 6-well cell culture plate with clean culture medium. Then place the plate in an incubator to culture the females until they release egg sacs. After the egg sacs are released, use a Pasteur pipette to remove the females from the wells of the plate. As soon as animals at the first copepodid stage, or C1, start to emerge, use a P10 micropipette to collect them. Every 2 to 3 days, monitor the wells for molted exuvi. After this, sex the animals by examining their antenna morphology.
To correctly identify adult females, make sure that they have already molted 5 exuvi in culture wells because differences in shape of antennae between adult females and juveniles are not visibly significant in some populations.